Now we officially start the tutorial session. So let's start with you asking question. Okay, so you have the mic. Yeah, go ahead.
Like you have said that like T01 knockout or DNMT3A knockout. Actually, like if we do the knockout, it is totally irreversible. And we
Yes.
know like DNMT3A or 1011 translocase 1, that
Yeah.
particular enzymes have a very over, like we can say like it has many different roles it plays. If we like totally knocked it out, then like
That is a very good point. So tell me, how will you switch it off without knocking it out?
like ma'am, it should be like in my perception it should be like reversible, not totally knocked out.
Yeah, so how would you make it? I agree with you 100%. How would you do it?
Knockdown can be possible, or small molecule inhibitors can be possible.
But if you are dealing with gene therapy, right? This is under gene therapy. How would you do that?
Yeah, like ma'am, RNA no.
Think about it. See, before I answer your question, let me say something. So this is always the case that people find some target like DNMT3A, right? Or calpain, that is a protease. So the whole world thinks, oh, we found the main target, let's knock it out, right? Or knock it down. And then what happens, they realize, oh, this has many good functions, and the whole body falls apart. So you cannot knock it out or knock it down, right? So you are absolutely right, because it has a lot of good functions too. So what do you do then? You can obviously use small molecule inhibitor. What else can you do? So in cell lines, it is easy to do reversible inhibition using small molecules. So you add it and it will get, so shRNA, siRNA, right? It is inducible also, that whenever you add doxycycline, it will be knocked down. When you remove doxycycline, it will come back. So you can do reversible. I don't think people will do it in humans that way. Okay, so unless it is safe to knock it out, you would not knock it out, you would use the inhibitor. But you can also think about epigenetically regulating the expression of TET protein, right? So TET is epigenetically regulating other things, but you can also epigenetically regulate TET itself also, right? So you can go further upstream. But this is a very good point. You just don't want to knock out just because it is doing one bad thing. You have to weigh the pros and cons of everything. And sometimes it is good to be reversible in that respect. So, yeah. Go ahead.
Ma'am, my doubt was, we talked about cancer vaccines, and in a normal vaccine the procedure is that you attenuate the causative organism to the point that it is no more infective, but it still has the characteristic protein expression that we need for the immune response. But in case of, if we are trying to do it for cancer, wouldn't it require a higher degree of attenuation, because cancer cells or tumor cells have many such things that could induce cancer as well?
100%. Huge amount of this thing. You have to really kill them so badly that there is no way even one single cancer cell is alive there, because otherwise we are introducing cancer there. So they have to be like really dead. And I would not be surprised if they mechanically lyse it up without destroying the proteins. So yeah, you have to do that. You don't want to introduce cancer there. Yeah.
Another doubt that I had was that we talked about oncolytic viruses. So you had told us that the viral DNA is edited in a manner that it does not, the infective parts of the DNA are removed and only the part that we need to introduce into the cell is kept. So how does our immune system respond to such a viral invasion? Does it have a triggered response, or does it not treat it as such?
So it has to be modified in such a way. So what you do is that you give it in very small amount, okay, and it is engineered. So you can even make virus coated in some nanoparticles, and nanoparticles are coated with, let's say, folate. So cancer cells express folate receptors, right? So nanoparticle is hiding the virus, or you can even make virus which are expressing something which is recognized by the cancer cells, and you don't give too much virus. So basically the virus will go inside the cancer cells, bind there, and it will infect it. Now I don't know what, so this I know very well. You know, virus have viral proteins and coating proteins and all of these things. So these things are known for decades and decades now, right? So it was discovered in 1980s. So we know exactly what to mutate, what to remove, so that it does not touch normal cells. Now imagine that one virus particle going inside tumor cell, but they are replication. They can replicate in tumor cells. So they are replicating, and the way they replicate is that they will form trillions of viruses within a cell, and the cell cannot, whole cell is completely dead by that time. So it will lyse up and just burst open, right? So then these trillions of viruses will come out, but they are next to the tumor, they are within the tumor, and they are infecting more tumor cells and more tumor cells, and in no time the whole tumor will be eradicated, right, in an ideal situation. So those viruses are meant to replicate only in the tumor cells. So even if you don't add any toxin to the virus, just by virtue of replicating, it is lysing the tumor cells. But then because it is lysing the tumor cells, all the tumor antigens are exposed now. So immune system will kick in. It is like a really hot thing for immune system. So immune system will start attacking. And the third thing is that you can always add further toxin to the virus, which is further released there. So there are many ways to do it. Yes. Go ahead, go ahead. You guys are the first time attendees here, right?
Yes.
Yeah.
Ma'am, how can we integrate any gene of interest into only cancer cell in vivo? That means how can we target only cancer cell in vivo?
Oh, that
if
is...
it is based on their microenvironment or something?
No. So that is actually super easy to do. What happens is that cancer cells have their own special things expressing at the cell surface, right? So folate is one of them. They express folate receptors, and the reason they express folate receptors, have you guys heard of tetrahydrofolate? You have heard, right? And why is tetrahydrofolate is so important? Because you remember it carries the methyl group, the methylene group, 5-10 methylene THFs, 5-methyl THF, 10-methyl THF. So it is a one carbon donor, you know that, right? So this one carbon donor is very useful for the synthesis of thymine, okay? And how will you describe cancer in the most simplest word? Uncontrolled cell proliferation. That's the first thing. Before even we say metastasis, we say uncontrolled cell proliferation. So if something is proliferating, DNA has to get replicated all the time. And if DNA is replicated, it needs a constant supply of nucleotides. So nucleotides are shared between DNA and RNA, right? They both use. But thymine is unique to DNA, right? So basically, when DNA is replicating, in addition to GTP, ATP, CTP, it also needs TTP, right? So it needs a constant supply of thymine. For constant supply of thymine, it needs tetrahydrofolate, which can deliver the methyl group. For tetrahydrofolate, it needs folate to start with. So cancer cells develop these folate receptors so that they can intake folate there. So we scientists have made use of this property of cancer cells. So folate receptor is just a receptor, right? But folate itself is a small molecule, very easy to modify. So what we do, Professor Venkatesh has worked on this thing during his postdoc. So folate, this is a molecule folate, you attach a linker, and then with a linker you can add anything of your choice there. So suppose you add a fluorescent FITC or rhodamine or anything there. So then it will come and bind preferably to cancer cells because they overexpress, right? And cancer cells will light up. So this has been approved in Europe already, that you can use folate coupled to fluorescent molecule, inject it in patients, and the patient's tumors will light up, and the surgeons use it while doing surgery. So it is extremely precise, correct? Now instead of adding a fluorescent molecule, you can attach a therapeutic drug. So it is walking around everywhere, but it is going to get concentrated on the tumor, and the therapeutic will be delivered to the tumor cell. So I just gave you an example of folate receptor. There are many, many different things which is expressed there. Now let me tell you something else. Have you guys heard of RNA aptamer or DNA aptamer? Have you heard of it? No. So aptamers are basically what they are, secondary tertiary structures of DNA and RNA. So this is not double-stranded DNA. It is just a piece of DNA, and you do what is it called, CellX. Have you heard of CellX? No, that's okay. It doesn't matter. You make trillions of DNA molecules with all possible combinations, right? With error-prone PCR, or you can do chemical synthesis. And then you take your target of interest and you do binding assay. Without going into details, you can find the DNA or RNA which binds specifically to some target. So if you look at the DNA, it is just a short piece of DNA with a certain tertiary structure, and because of the certain tertiary structure, it is binding this very tightly. So those are called aptamers. So you can even take a DNA aptamer once it is recognized. You synthesize it in a chemical synthesizer, or you do it yourself. It's not a big deal. You synthesize it, you attach your thing. Every time, every single time when you have that sequence, it will fold in the same way. You know that, right? It's like the protein. If it has the same sequence, you can make it in the universe anywhere I cannot say anywhere in the universe. I don't know what happens outside of Earth. But on Earth, anywhere you make it, it will fold in the same way. Same thing for DNA aptamer. So once you know the sequence, you synthesize it, you attach it to anything you want, and it will bind to the cancer cells. So basically, you could even take cancer cells and use it for screening to make aptamers, even if you don't know the receptor or anything. So what you do, you take normal cells, you take cancer cells, and you can see which DNA binds here but doesn't bind here. So you can select it. Once you have that tool, you can attach anything you want, fluorescent molecule, drug, radioactivity, toxin, whatever you want. It will go to the tumor cells. Then you can also do this thing that you can take nanoparticles, right? You can make a nanoparticle. Think of nanoparticle like a box. You can put anything you want inside the nanoparticle. Outside, you put those aptamers. It will be delivered to the tumor. So let me tell you one more thing. So you guys are very worried about the tumor microenvironment, and you should be worried about the tumor microenvironment. But there are some tumors where tumor is more accessible than other tumors. Like pancreatic tumors are not accessible, but other tumors are accessible despite the microenvironment. If the microenvironment is very acidic, you might want to consider really using nanoparticles or something. But anything cancer does, we make use of it. Yeah, I think it is in one of your lecture, in this one also. I teach so much that I'm confused where I've included that thing. So see, you guys can really enjoy this thing. So microenvironment is acidic because it releases acid. Why it releases acid? because the immune cells are ineffective there. But now we know you guys, like cancer cells are secreting acid. So now we make a linker which is pH sensitive. So it is it will not fall apart anywhere in the body except in the tumor microenvironment. When it falls apart, stuff gets inside the tumor. So to dal my path. So whatever they do, we find the answer to that. We make use of their own thing and attack that way. Answers your question?
Yes, ma'am.
Yeah. All right, guys. All I can see is that after lunch, at least everyone is awake. We needed that break, right? Go ahead.
So, ma'am, is oncolytic virus therapy only one-time therapy because our immune system will identify the virus and next time it will act as a vaccine kind of way, and our immune system will identify the virus for the next time and it might get killed earlier before reaching the tumor?
That is a good question. I would think that it should be a one-time therapy, but the problem is that the cancer cells keep mutating. So even if oncolytic virus attacks, there is a chance that some cells survive and then they will mutate it, and this is a very well-known fact. If you make anything against a known antigen, they will hide that antigen. So they will not express that antigen and the immune system will be fooled. So, even for COVID, right, the virus is mutating. That's why we keep getting vaccines, I also got an alert in my phone, come for the vaccine thing because COVID cases are at rise again and virus is mutating. So whatever vaccine we have already taken, we have immunity. If COVID comes in that form, we don't have to do anything because our body will fight it off. All the B cells are there. But if COVID changes the whole spike protein, and if our body has never encountered it, it's again a new story, right? So viruses mutate very fast, you know that, right? So do cancer cells. So whatever is the recognition element which got them attacked, they will make it disappear. So it is just it is a constant fight with this thing. Having said that, many cancers are eradicated. I know of a person very close to me in my family. So she got extremely severe cancer and it is completely eradicated.
Virus which we are using, so if we are going to do the oncolytic virus therapy second time, we will need to change our virus too, na? Because our virus will be recognized by the immune system. So before reaching the tumor, our virus will get killed by the immune system because it's a foreign particle.
No. So what you do is this. You first of all, you don't give a very high dose of the virus, and you can make the virus pretty anti-immunogenic. You can hide the virus, you can like wrap it up in a nanoparticle or something. So virus itself is not triggering the immune system. Okay, it is that when virus gets inside the cells and start replicating, that's when the whole cells fall apart. So virus you can make non-immunogenic. See, you guys know that antibodies are raised in rabbits, polyclonal. You all know that, right? So long time ago when I was a postdoc, we wanted to make an antibody towards a target I was working on, right? So in those days, antibodies were not that easily available like they are here. So I wanted to make it. So I was a chemist, so I made the whole peptide, synthesized the peptide myself. We didn't have a synthesizer. So I don't know how long it was, 10 mer, 20 mer synthesized. And that was against my protein, right? So I made it. But if I inject, I had to send it to some company for injection. But if I send the peptide and they inject it, it will not trigger immune response. So I had to couple it to KLH and do something so that it actually triggers immune response. So you can make the virus so that it is non-immunogenic, like in some way. I don't know which way they do it. So that itself does not do the immune response. So keep in mind, when you give the oncolytic virus the very first time, immune cells are only coming after the tumor cells are lysed, and immune cells are coming against the tumor antigens, not the virus. Make sense? And if virus should trigger an immune response, because COVID virus triggers an immune response, right? So the chances are that they change it in such a way that it doesn't trigger an immune response, or they package it in something.
I have one more doubt. How does ubiquinone how does ubiquitin ligase identify the protein of interest?
So what happens is this. So I will tell you about the ubiquitin system not tomorrow but on Monday, because tomorrow we don't have any class. So there are three enzymes, E1, E2 and E3 enzyme. Okay. So E1, first the substrate will bind E1, E1 will come, then E2 will come, then E3 will. E3 is the biggest one. Okay, so E3 is the biggest one. So on one side it will recognize the substrate, and E1, E2 are all there. So then it will, so it is, when the whole complex is formed, it is the E3 which recognizes the substrate. So you have a lot more E3 than E2 and E1. So E3 is like a big adapter protein which is bound to E2 and E1 and also the substrate. And it is the E3 which is recognizing the substrate. It will not make sense till I show you, because it's the whole ubiquitin machinery. So I have to show you the whole thing before you understand what I'm saying. But the bottom line is that when you make PROTAC, you have two handles. One part recognizes the substrate, one part brings in that machinery. And once it is in close proximity, ubiquitin tag is added to the protein of interest and it gets degraded. It is very easy to simplify, to understand once I have all the images there. Because many times only hand waving doesn't help, you know. So you have to see the actual picture. But thank you for asking me, because I will now make sure that I have, I don't have those, I have a lot of figures, but I will go even more basic to explain you simple ubiquitination mechanism independent of PROTACs first. And I have it everything at home, but I obviously have not brought it here, but I can pull it out of the web and show you guys everything. I am supposed to ask you some questions too. Yes, go ahead while I'm thinking.
Ma'am, if a person is reacting to a drug, harshly reacting to the drug while others are taking it easily, how do we identify, is it because of genetics or epigenetics?
If it is due to epigenetics also, it is affecting the expression level of proteins, right? Actually, you have asked a very good question. I will open the floor for everyone to answer this question. Okay, so you pose the question on my behalf. This is a really excellent question. Can you repeat it? And let's see what you guys come up with.
So, if a person is reacting to a drug harshly while others are taking it easily, how do we identify if it is genetic or epigenetic?
That is very, very, very good question. I mean, that is just really fabulous question. Think about it and tell me. Yes. Rohit. Okay. If I remember tomorrow, then I will pat myself.
Like ma'am, for genetic cases, we will first like target the genetic. We will do PCR for particular receptor if it is expressed for the drug. So
So what kind of PCR will you do if you have to see whether it is overexpressed?
like normal PCR, real time PCR.
Real time.
Yeah.
Very good. QRT PCR.
Yeah.
Very good. Yes.
Real time PCR. If it is like normal in case like genetic after doing real time PCR, like if it is normal, then we will go for like epigenetics. So we will do attack sequencing, or we
Chip
can
seq
chip
sequencing.
sequencing, attack sequencing to
But how will you? So here is the thing. How will you know there are 30,000 genes? Where will you attack? Like where will you focus?
yeah,
How will you?
like promoter region of the particular gene, which is
particular gene you will go after?
particular receptor for that, like it is responsible for the drug, like
No, so see, the question is you are Venkatesh, right? So the Venkatesh's question is that if a person is reacting harshly to a drug, so keep in mind he's saying harshly to a drug. Okay, so that means we have already decided the drug, and if we have already decided the drug, we already know the receptor, right? But he's reacting harshly to the drug. So do you think it is because of the receptor? All of you, it's open to all of you. This is actually such an excellent question because it will cover everything that we discussed today, every single thing that we discussed today, right? So let's go back to HER2 because every time we go back to HER2, right? So we are doing HER2 here, okay? So we know this patient expresses HER2 receptor and we are using Herceptin or we are using Lapatinib. There are many HER2 receptor things, right? So we know the receptor, okay? We know the target. But if it is, what are the possible reason for the patient to have an adverse reaction to the drug that we are administering? What could be the possible things? Let's make a list of those and then we attack that thing. Can you guys tell me? So do you think, is it because HER2 is overexpressed or HER2 is not expressed? Even before you do qPCR, what is your hypothesis? No, you tell me, what is your hypothesis? Do you think the patient is having nausea and all of these things because of HER2 receptor? So here is the thing, if all the patients across the board are having bad this thing, you can say the drug is toxic, right? But someone is having toxicity, others are not, right? That's what you're saying. So what would you think? What is going on? Shivani, you want to try? Hyperimmune response, why? Because others are not having hyperimmune response. I'm going to be the devil's advocate here, so don't take it personally.
It can be because of metabolism as well.
It could be due to metabolism. So we have to get out of the targeted therapy right now and we have to go to the metabolism. So now you have guessed it is due to metabolism. What would you do? You were going to say something, right?
It could also be due to the uptake regulation, like if the drug uptake is not proper.
Please, you have to use it. It has to be recorded.
It could be due to the drug uptake as well if it is not being uptaken properly, if it is too slow and the amount is being accumulated too much and there is a toxicity due to that. That could be also the case.
That is also the case. So it could be drug uptake issue, it could be drug metabolism issue, it could be anything due to all the PGX we said. So now you have narrowed down it is due to PGX. What would you do? What will you guys do?
Like as we do that study, which enzymes are like not playing their role, like analysis in pharmacogenomics, what we learn.
Yes, so there is
Cytochromes.
already a list of like 20-30 genes. We will check those
You
genes.
can check transporters and the metabolizing enzymes as well, transporters and all that is responsible for pharmacodynamics and kinetics.
So we will do QRT-PCR and we'll look at the levels of those things. And what if all the levels are equal, then what will you do? So between normal and this patient, they are equal, then what will you do next? Tell me. You have to use the mic.
So, if normal and the other guys' PCR result are same, then we can conclude it is epigenetic.
No. Because epigenetics is expression of gene, and if it is equal, then it is definitely not epigenetics. You will rule that out, right? Because epigenetics is gene expression. Correct? So you have ruled out epigenetics here. But then what next? Huh? So, I want you to think before I say anything. So DNA makes RNA, RNA makes protein. So you did qRT-PCR and all the genes are expressed at the same level, right? So, and epigenetics is controlling gene expression. It seems they are the same. Hey, don't hesitate to say anything because I may be missing it. If something is, just say it. Do you think epigenetic could be playing a role? Because I'm also thinking as you pose the question, right? Don't be afraid. We are just
Yes,
throwing ideas here.
I'm not sure about it, but ma'am, when we do RT-PCR, our final result is sequence. If I'm going in right way, the DNA sequence, I guess.
Very good.
So
I
if
was
they are
hoping
same,
somebody says that. So if qRT-PCR results are the same, then you also have to do DNA sequencing. What if RNA levels are the same but the proteins are mutated, and if the proteins are mutated they're going to work differently. So you will sequence the DNA, right? You absolutely need to sequence the DNA. So suppose the sequences are wrong, then you have answered your question. It is at the genetic level, not at the epigenetic level. Now let's go further than that. What if your DNA sequence is also identical? What will you do then? You have asked such an excellent question. I don't have to plan anything because the whole thing is covered in this one question. Tell me, so DNA sequence is identical, correct? What is it? DNA sequence is identical. Epigenetics is not very important role, or at least because expression level is the same. So what else is going on?
Maybe at the protein level.
At the protein level. So what would you do? Very good. So what would you do at the protein level?
We can check with the immunoblot techniques.
Exactly. You will check it because it is only 20-30 proteins. You definitely can do with the immunoblotting, and it is entirely possible that the RNAs are equal, proteins are not equal. Definitely possible. Why? Because protein degradation rate may be different, right? Then protein modifications could be different also, right? So then you will go to the check at the protein level, and the chances are that you will find the protein levels are not equal. Okay, now let's assume that the protein levels are also equal. Now what will you do? So obviously there is a scenario that we have not yet discovered that gene, which gene is playing a role, right? Because we are focused on these 20-30 genes thinking they will answer all the questions. So that is always a possibility. But before you throw out these 20-30 genes out of the picture, right? What is the one other thing you can do which will conclusively tell that if these genes are involved or not involved? In that case, you will make the protein, isolate the protein from these cells, not make it, and look at their activities in vitro, because their activities may be different. So you know what? You may argue, well, if there is a mutation, their activity are going to be different, right? But your DNA, so if you see mutation, then obviously you will go and check the activities, right? Because with the mutation, the activities are going to be altered, correct? But in case the mutation has not shown you, the DNA sequencing has shown no mutation, but the activities are different. How is that possible? So that would be, in that case, that something else in the cell is impacting this protein. And there is another player involved which is controlling the activity of this protein. Maybe it is binding, maybe it is attaching to it, or maybe something, or maybe it has changed its localization or something, and that way it is altered. So to sum it up, you will look at the RNA, you will look at the DNA sequence, you will look at the protein levels. After all of this, you are going to check the activity of each of these enzymes. Because if you remember, activities were different in each case. Usually it is, so in the case that I showed you, they were mutated at the DNA level. So QRT-PCR would not have told us that much, but having said that, the levels were also different. So QRT-PCR would have shown at the RNA level, but not at the protein level. So you have to look at QRT-PCR, you have to look at the protein level, you have to measure the activity. And if all of these things show nothing, then there is something else involved there. Okay. Now, finally, I come back to the target. Okay, so let's say you find nothing going wrong, okay, and the patient has a lot of toxicity. So what else could be wrong?
It can be liver function.
It could be liver function, but now I am actually going back to where you started, the target. So it is possible. Well, I am assuming that the person has already sequenced the target and has ensured that the target itself is not mutated, but there is a possibility that this patient who is responding badly, the target has been mutated and it is no longer binding the drug. So as it is getting exposed, so now I am getting back to the target, and because it is not binding there, it is not other than the metabolism, it is like statin, it is not being used, and so I don't know, the target itself could contribute in some way, because it is not like other people, they are getting better, this person is not getting better because drug is not engaging with the target. So that could be the toxicity issue, because others are going in the right directions and he is deteriorating, or he or she is deteriorating. Cancer, right? So you have to figure out why this cancer occurred. Is it a viral infection, right? Sometimes it is at such an advanced stage you don't have time to do all these things, but you have to find. In case of liver cancer, if specific types are known which express receptors, you have to test quickly for all of these things to ensure that, right? And then once you know what is the best therapy that you can give, then you go for the targeted therapy as the first choice. If that is not a choice, then you go for immunotherapy and chemotherapy. If surgery is a choice, you go for surgery, if it is a choice. In US, in few places they do something called histocrispy or something like that. I've heard it recently. So if somebody has lung metastasis, they can manually go and clean up that thing, but it is very complicated process. So they do it under certain circumstances, like when the patient is recovering, the patient is responding to chemotherapy because they are afraid that they will do such an invasive process and the cancer will spread anyway. So when cancer comes under control, they think they can do it, but it is still pretty bad. Yeah. So there are ways, but the bottom line is that if it is not good today, it will get better tomorrow. In the last 10 years, we have made miraculous progress. Like I was telling you about this faculty member who had cancer, right? 2-3 years ago, and he had to sign so many paperwork for 3 weeks before he was given immunotherapy, and it was too late by that time. So, and now it is given worldwide. So it is very rapidly increasing. You saw how, and immunotherapy is biologics, right? It's PD-1 and VEGF inhibitors antibodies. So these are antibodies. And all major cities have these facilities here in India. I don't know about rural places, I have no idea, but I know major cities have. One last question before we can go for tea. Do you guys want to continue after tea? We should continue. We should continue. Any other question, anyone? Yes, yes, go ahead.
Ma'am, actually, I came from pharmacy background. Actually, I have a confusion. Like, I get confused when these things, like you said, immunoblot, then RT-qPCR. So we didn't explicitly study these things. So I get confused. Like, what is the mechanism? Can you briefly tell me what it exactly gives you, like results? That day also you mentioned that RNA chip and all.
Yeah, absolutely, absolutely. So let me ask you something. Do you know about, suppose you have to, why do you even look at the RNA levels? Why can't you look at the DNA itself? Can anyone tell me this thing? So when you're comparing two parts like normal cells and cancer cells, why do you look at RNA? Why don't you look at DNA of normal? Why don't you look at the levels of DNA? Huh? I am asking you such a stupid question. You are answering it very, in a very reasonable manner. You are thinking my question is genuine. It is not a genuine question. It is actually a pretty stupid question, and I want you to answer it in that way. So don't explain to me. Don't think I'm telling you the right thing. So, because I'm asking you really weird question, the weirdest question I've ever asked. Hey, anyone tell me, why don't you measure the DNA levels instead of RNA levels? Why do you have to go to RNA levels? But DNA is the father of RNA, so why don't you look at the DNA levels?
Because it's the expression we check, like how much it is expression.
Yeah, so why can't you check the expression level of DNA?
DNA is like it's present in the genome. It may express, it may not express.
That's the thing. DNA may express, may not express, but even better answer is what? DNA levels will never change. DNA is DNA. It is just that one copy there, haploid or diploid, right? So there is no DNA level, right? Okay, and second, this is first thing, there is no DNA level. DNA is always exactly the same in every cell of our body, first thing. Second thing, as you said, 5% of the DNA will make messenger RNA. So why would you ever look at DNA? It makes zero sense to look at DNA, correct? Yeah, see, so I have to trick you. You have to answer me with the same thing. Okay, you cannot test. So, okay, sometimes you have to throw this thing. RNA, okay, so we cannot check DNA. We have to check RNA. So why can't you just take out the RNA from the cell and measure it? Can you do that from human cells? Can you take out the RNA from the human cells? No. So let me say something. Yeah. Anyone has done QRT-PCR here? You have done. May I know your name, please? Enos? Evans. Can you spell it for me?
EVANS.
Very nice. Evans. Okay. So Evans, when you do QRT-PCR, can you tell me how you do it? It is 3 o'clock. Why don't we pick it up? You guys discuss it, because this is very interesting question. You will learn a lot about it. No, it is not. No. If you do clinical trials, if you read clinical trials, a large percentage of patients do not receive the drug. A large percentage of people, they are deliberately given sugar pills, but they don't know they are getting sugar pills. They believe that they are getting the best medicine in the world, and they get well. And the strange thing is that those who are actually getting, some of them do not get well. And this thing has nothing to do with drug metabolism or anything like that.
Cancer cells have something to do with pluripotency.
Pluripotency, no. No, pluripotency is a feature of stem cells. Yeah. Cancer cells are not pluripotent. They are not.
But when you discuss about they turn into mesenchyme.
Oh, that is different. That is different.
They change their like physiology, right?
So they overexpress those proteins which make it mesenchymal, but they are not changing their lineage. They do change their lineage also, but they are not called pluripotent. Pluripotent definition means that they can change it to so for example, castration resistant prostate cancer cells, if you give them androgen receptor therapy, they become neuroendocrine prostate cancer cell lines. So they have changed the lineage, but you would still not call them pluripotent cell lines. Pluripotent are those which can differentiate into many different cell types, and they will function as those cell types. Cancer cells are just adopting a new phenotype to survive. They are not serving anything, you know. They are not becoming endocrine cells and secreting something. They are just hiding in plain sight.
So they have nothing to do with acquiring different spaces and acquiring their functions intentionally and doing the process as same as the normal cells do.
They are not doing that.
Okay.
They are just trying to survive.
Yeah, all they do is just take up all the nutrients what they
Yeah. It is, do all the damage. I have never seen that they are functioning for the body.
Okay. Is there any way that if this is the cancer, this is the microenvironment, right? Is there any way we can just shut it off? We isolate this section, even if it's in vivo. Can we just
Seal it off.
remove all the access to the nutrients?
That's what you are doing with the angiogenesis inhibitors, right? That is exactly how it works. So all the nutrients are going through the bloods, right? Oxygen is going through the blood. So you are cutting down angiogenesis, and that is very effective. Okay, but then some cells survive even under low deprivation condition. They cannot survive without glucose. See the tumor, so suppose this is the blood vessel, the tumor will not come here because here there is no glucose. Oxygen comes up to this place, glucose comes up to this place. So they will stop here till they make new blood vessels, then they will grow like this, right? So that is why they make blood vessels. So when you cut down the blood vessels, they shrink, they always shrink. But then what happens is that some cells survive under hypoxic condition. Of course, they are eating glucose, they don't have enough oxygen, but they become extremely resilient, and then you cannot kill them. The cells which survive under hypoxic conditions are extremely resilient, extremely resilient.
Ma'am, at what stage of therapy we think about giving angiogenesis inhibitors?
You give right away. You give right away. The moment it is diagnosed, you give anti-angiogenesis.
Like we know that it is metastasized, then only
When it has metastasized, you absolutely have to give that, because once it has metastasized. So tomorrow I am going to tell you about chemotherapy, surgery and radiotherapy. We have talked about precision medicine, now we'll step back and tell you the conventional ones. So when it has metastasized, you give chemotherapy absolutely and angiogenesis inhibitor therapy, because now you cannot do surgery. You have to give systemic therapy. So you definitely give immunotherapy. For my friend, it was localized, right? Even then, he was given immunotherapy because immunotherapy is very effective. When I was preparing the lectures here, I came across a paper which said that immunotherapy works in 20% of the patients. I was surprised to see that, because I have seen it working in huge number of patients. The only problem is resistance. So That is why I think, this is my thinking, that if one has to tackle cancer, it has to be very quick and very before it develops resistance.
Is there any certain period of time after which it, for every cancer, does it vary?
No, few cancers can mutate very fast, right? Others are not that quick. But it is a very weird thing. Sometimes slowly growing cancers are more aggressive. The fast growing cancers are less aggressive. It's just a very, there are many factors in play to determine which are aggressive. And like EGFR receptor, right? So HER2 is a EGFR family receptor. So EGFR receptor is upregulated in lung cancer, in breast cancer, and in many other cancer, right? So EGFR is very easy to target using small molecule because it's a kinase, it's a nice pocket. So they were given, so then it developed resistance. So then the second generation was given, it developed resistance. Now the third generation was given, it was highly effective, but then it developed resistance. So how does it develop resistance? So you inhibit EGFR, so EGFR is inhibited, so now they will upregulate another pathway, bypass. So EGFR is no longer needed, this other pathway takes over. So once you figure out again second pathway is taking over, then you combine it with this guy inhibitor also. So then it works for us time. In cancers it is, I mean in some cases it is obviously it goes away completely. In other cases it is a playing game, how long you can prolong the life. Yeah.
We discussed about removing a lot of receptors from T cells.
Say that again? Oh, you have to speak up.
We discussed about, is it okay? Yes.
No, it is not.
Okay. We discussed about removing a lot of receptors from T cells before, to
So that it is not recognized as a foreign
particles, yes. We were just inhibiting the host and the donor interaction in an aggressive way so that they don't counteract with each other. So then will it not affect the immune aspect of the T cell?
No. So it is the things which are removed, you keep the power of T cells intact. So what you do is that is something will come obviously next week because it is the final week. So these T cells, when you put these chimeric receptors, they are not alone. So I am going to show you a slide like first generation CAR, second generation CAR, third generation CAR, fourth generation CAR. So now I think 6-7 generation CARs are there. So you have these receptors, but along with these receptors also you have specific proteins co-expressed with them. So they bind, they get activated, some release cytokines, some they. So there is a whole army of things. This is a completely engineered thing, okay, based on how T cells react. So this whole generation of things have been added to that. And there is a lot of, people do a lot of fun stuff with CAR T, like they will make it so much fun, like different kinds of chimeric receptors, right, in one T cell. And also, so it is completely equipped, like you are sending a person in the war zone completely equipped with all kinds of weapons. But you make sure that they don't, you are fighting for the host here, right? So the host cell doesn't attack you. So you have to be very friendly with the host cell and not attack the host cells and not get attacked by the host cells.
So are we giving immunosuppressors also?
No, we don't, because if you give immunosuppressor, you are shutting down the immune system, and then these test cells will be also shut down. You don't have to do that.
That means they're enabled with the power of killing.
Yeah, they have to be. Otherwise, CAR T cells are very, very effective in killing it. And then there are these NK cells, you know that. So they are CAR NK cells also. Yeah, those will also come there. So they have their own power and they have their own limitation. So people use CAR NK cells also.
This CAR M cells.
There is a CAR M cells also, the macrophage version of it. Yes, CAR M cells are there also. People are having a lot of fun. doing, once a new technology comes, they're adding it to all different kinds of cell lines. So all of these things are in your lectures.
Can we predict the change which might, can we again
Predict the change what cancer might have in the next mutation.
So usually, so I'm going to show you a slide probably on Monday. So you can and you cannot, both things are true. So for example, when Gleevec was given, right, so there was a huge celebration at Novartis, and while we were still celebrating, right, when the first patient got it, the celebration was not even over. We had news that the patients have developed resistance, right? And I will show you a slide in which it shows at least 20-30 different mutations which occur due to Gleevec treatment. Okay, so yeah, we could not predict which mutations will occur. Okay, so there were so many mutations. So that is why there is new generation of Gleevec, like modification. There is one way you could possibly predict is that what you do, you take a cell line, right, which is basically, which has BCR-ABL in it, right? Keep in mind, this is a cell line. So you take as close of a cell line like CML, okay, with BCR-ABL in it. And you treat it with Gleevec at a very low dose, which is non-lethal dose. And you do it for 3 months, 4 months, 5 months. And then it will become resistant to Gleevec. And slowly you keep on increasing the dose till it is still remaining non-lethal. And if you sequence it, or if you do immunoblotting or anything, you will figure it out what proteins are upregulated. So that is a way in the lab that you can see what are the pathways which are upregulated. So you will get a pretty good idea before it shows up in patients. But keep in mind, human body is more complex than a cell line. So it is, yeah, so it is hard to predict what else. But also keep in mind that the PROTAC thing is there. So you can, people have made Gleevec PROTAC. So PROTAC is ubiquitination machinery on one end and your target in one end. So if you have to bring them together, so how will you bring kinase close to something? You have to target the kinase pocket, right? So on one end you will put Gleevec for BCR-ABL because that is going to bind BCR-ABL. Then you will put a linker, right, to give flexibility. And then you will put something which binds to the ubiquitination machinery. So you will make a bifunctional molecule, that's PROTAC. And you give it, and then it will bind both things, they will come together, and once they come together, they will get degraded. Right? So in that case, even if it has all these different mutators, you are just getting rid of the whole thing. But it should still bind to the pocket enough to get degraded. But if the mutation prevents it from binding it, then there is a problem.
But you said cancer behave different in plate and different in your human body. What if it doesn't mutate exactly what you have anticipated?
That is likely, but at least it will also give you some information, which may be helpful. Again.
Do tumor have any sort of half-life? Have you ever seen? Because we have seen in lab, they do die on their own after some time.
Tumors don't have a half-life because cells are dying, more cells are getting. It is always on the rise.
Their balance with dying and rebirth.
Yeah, so balance is always towards more proliferation, less death. So
my question was, even if we could calculate the half-life just in case. So we could make it stationary for a while so that we are giving our treatment to it till it is at its
People have done that. There are two things you can do. Some things will arrest cell growth. These are called cytostatic. And other compounds will kill the cells. These are called cytotoxic. So yeah, people do that all the time. The thing is that it is hard to
maintain that state, right?
yeah. Go ahead.
Ma'am, we have discussed about biologics and biosimilar. So basically, the biosimilars are mimicking the action of biologics only,
Yeah.
and they are the cheaper
Yeah.
alternates. So my question is that can we use biosimilar in place of biologics all the time? Means the efficacy will get hampered or not?
No, efficacy should not get hampered. If efficacy is hampered, it is not a biosimilar. Because if you're treating something, if it is like 2-3%, maybe the price is very cheap, then maybe somebody will go for a biosimilar. But if it is 20% less, nobody will use a biosimilar. So biosimilar is not a biosimilar if it is not similar in efficacy. Then it is a cheap something. It is not worth anything. It has to have the same activity. So you can think of it as a generic drug versus the branded drug. They are identical. At least in chemistry, they are identical, right? But in biosimilar, it would be more or less identical with minor modifications. Suppose you know something works, right? But you also know if you attach this thing, it will become more stable. So those kind of modifications may be there. Maybe a cheaper version, cheaper way of making it. So. Because if you make a biosimilar which is not as efficacious, biosimilars are usually given for pretty serious diseases. Biologics are not given for fun, right? Because they are so expensive. So it has to work very efficiently, especially when someone is dealing with a disease like cancer which mutates. So you don't have, every day counts. You cannot give cancer time to mutate. When you guys take antibiotics, right? So when you take antibiotics, the doctor gives you, is antibiotics, can you buy it in the stores these days? No, can you buy in the stores these days? I'm asking you. You cannot buy in the stores these days, right? You have to have doctor's prescription. In my time, it was freely available, right? So people used to buy, I have myself bought antibiotics, like tons of antibiotics. Every time you do this, you take antibiotic. Because, so then what happened, that we read it in a newspaper that a mother was giving antibiotic to her son who was 3 months old. So by the time he was 6 months old, he was resistant to antibiotic, and then he really had a bad attack of something and antibiotics did not work and he died, right? That was what opened up this thing that antibiotics cannot be given randomly, and that's how they became a prescription drug, right? So having said that, when you are given antibiotics, suppose you are given 30 tablets or capsules for 10 days, do you take the whole thing, or do you stop in between, or do you miss the dose? Even if the disease gets better. You know that, right? It is absolutely essential, because suppose I can miss some other drug, I cannot miss antibiotics, because what happens, if you miss antibiotics, you give bacteria a chance to mutate. And if bacteria mutates, we are in serious trouble because it is resistant to that antibiotic. So while you are taking antibiotic, you cannot miss any dose. You cannot miss any dose. You cannot give bacteria a chance to mutate and attack you while. So you are taking antibiotics so quickly that they are dying. They just don't any time. Any one residual bacteria is also dead, right? That's the whole principle behind it. So you don't want to play with cancer and give it time. You don't want to miss anything.
Ma'am, after knocking out TCR in the CAR T cells, it cannot differentiate between self and non-self with the host cells. So how will it differentiate the cell? CAR T cell will differentiate between tumor cell and not self
because
cells?
they are expressing these chimeric receptors, right? So the TCR, which would have attacked the host cells, is gone. So, but they have this chimeric antigen receptor, right? That is designed towards the host, that is designed towards the tumor cells. So they will only attack the tumor cells. Is that your question? Right? And it is not that simple either. So when I tell you about CAR T cells, so there are several generations of CAR T cells. So initially they just put the chimeric receptor there, but now they put a lot of other things there, like CD28 or CD something molecules. So in actual T cells, when the receptor is there, the CD28 or whatever it is next to the receptor. So when the regular TCR gets activated, it is bound to this one and this secretes something. So they have mimicked the whole structure so that it functions like a real T cells, releases all the things. And what T cells do, they release this thing called granzyme or something and perforin when they are activated. So when they lock horns with the tumor cells, they release that, and that goes and it creates hole in the tumor cells, and stuff gets in and kills the tumor cells.
I have a basic question. Like according to all the like therapeutic strategies we have discussed, so among all which one is the higher, like which treatment has a higher chance of relapse, like according to all?
I don't know that thing. I don't know that thing because it totally depends on the cancer, kind of cancer, and what stage it was diagnosed, right? And so it is something totally dependent on the kind of cancer, at the stage it was diagnosed, and then whether targeted therapy can be used. So it totally depends on the treatment plan also. So, and then it depends on the person's physiology, it depends on the person's microbiota, person's health otherwise, because to fight cancer you have to have healthy body. So for prostate cancer, right, prostate cancer is not that bad of a cancer, but when it occurs in real elderly people, you give chemotherapy, they have no strength to deal with chemotherapy, so it's fatal for them. Just chemotherapy is fatal. So it depends on many factors. But younger people usually recover faster, but then younger people also die because if it is aggressive. So, and people with all the money in the world cannot fight it if it is aggressive. Like really famous people with, super famous people got the cancer and they could not fight it off. So it is a pretty bad disease. But the funny thing is that we didn't hear about this, I don't know, long time ago we never knew about this thing. I don't know whether we did not know about this thing or it didn't exist, because it seemed like it is a relatively recent disease. Part of it is that we are also eating a lot of processed food and things like that, which is also contributing to that. Initially we were using all raw food and raw things, so.
The question is similar to that. Ma'am, what do you think combination of food, like a bad combination of food, does it leads to cancer? Because in Ayurveda, they do believe in eating subtle food with certain combinations, while in historical way of eating, when we don't combine, which is oil soluble and which is acidic. So we can't eat something tartaric with combination of milk or something. We can't.
Yeah, I have heard of that.
Yeah. What's your take on that?
I don't know much about that. The way I eat is what I like to eat, kind of thing. So, like for example, I know you are not supposed to eat yogurt at night, right? But if I have to eat dahi bada, I have to eat dahi bada, right? So I know I should not eat yogurt at night, like kheera, cucumber. There is a saying about cucumber, if you take in the morning, it is hira. Kheera is hira in the morning, like if you take it at breakfast. In lunch, it is not as good, and it is almost zahar in the evening. But if I have to take kheera, I will take whenever I feel like taking kheera. So I don't follow these rules, but I'm 100% sure these rules are really important, because Ayurvedic is extremely important and nothing is wrong there probably. So I would respect Ayurveda over my own knowledge about food. But whatever they say is correct.
We are seeing the more disturbance we are caused with our diet, the more we are moving towards acquiring cancer.
Oh, there is no doubt about it. There is no doubt about it. And one thing I can share, this is my own experience. So it happened to me in India also, it happened to me in US also. So I was in Gyan course last time, right? So after the Gyan course, and I'm going to the same restaurant again this year also. So we went to Sayaji or something, right? So that was the last day, and it was 24th December that time, because I left on 25th December that in that Gyan course. So 24th night, I went to Sayaji along with the whole group of people, and I ate food there, and I was so sick on 25th. My flight, I mean, it was horrible flight for me, because it was just as bad as it gets. I somehow landed to Chicago, and there was no way I could. My home is 3 hours away from Chicago, and I was in no way to go there. It was just a long story. And why I had that reaction, the food was very tasty, and then I realized that they had added preservative, because they had about 100 different variety there. Honestly, no joke. So if you make these many food, you cannot throw it away. So they are preservatives. So they freeze, they thaw, they freeze, they thaw, they freeze, they thaw. And I, whatever my PGX is, I cannot deal with preservatives. So that had the strong reaction, right? It took me some time to understand what, because I didn't eat much and I was so sick. So this happened in India, and this happened exactly to me in US also. So again, I went to an Indian restaurant there, and they make food like buffet, day in and day out, evening, lunch, dinner, lunch, dinner, lunch, dinner. And I ate there, and I had taken a speaker. That day was Shivratri, and I had taken a speaker for dinner. And we were eating, and I got so sick that I had to lie down on floor. Like, I thought I will lose consciousness. So my husband came, and he took the speaker and dropped him to the hotel and took me to the hospital. And again, it was the preservative thing there also. Like, while eating the food, I had such a strong reaction. So I know it is preservatives. And there is yet another story about the same restaurant. So when these people, they buy processed food in tins, you can imagine how much preservative is in tin food, because it is at room temperature, right? So that is actually poisonous to eat that kind of food. So, and then people drink soda, and they drink and they drink and they drink. Like, we are taking this kind of tea with so much sugar. Tea is not bad. This much sugar is also really bad. Like, maybe not for you guys, because you guys are going up and down all day long and you're burning it off. But in general, it is pretty bad to have this much sugar. So everything contributes. My grandfather never got anything like that, because the lifestyle was different that time, right? None of my ancestors, nobody had ever heard of cancer. So it is just the lifestyle. Then pollution is playing its own part here also. You haven't done anything wrong. Like, you are just living, we are just living in those conditions. And also, when you watch cooking channels or something, I'm just surprised, like processing. You boil aloo, then you make pakoda out of it, then you slice it up again, and then you make puri out of it, and then you take the puri and then you put it in a mixer and make a powder of it, and then you add the powder to that. I mean, after a while, I thought, how much more process? I'm not joking. I can tell you the website also. So even just because they have to make money, so they have to put a new recipe every single day so that they can keep getting enough traffic. So they are just taking the same thing and processing and processing and processing and processing. You cannot do that. When you cook food, you have to cook food so that the nutrients are preserved there. You guys are too young, but when pressure cooker came, that was the biggest selling point, that it preserves the nutrients because it is so quick. Otherwise, without pressure cooker, you're cooking forever, right? And that kills the whole thing. So when you make any sabzi, like dry sabzi, you don't add water and burn it. You don't add water. You let the water in the sabzi to cook by itself by keeping it low flame so that you preserve the nutrients, right? So what I'm trying to say is that our lifestyle has created these diseases. It wasn't there before, at least I have not heard of. But also there is another flip side to it, right? Earlier, when I was young, in India, the average life was 61, 62. If people reach 60, people barely lived beyond 60. There were always some exceptions. Then the median age increased and increased and increased, right? In US, it is, 85, 90, something like that, which is higher than here. So if the age is increasing, so are the diseases. If you are living up to 60, you're not likely to get many diseases. But then why would you die at 60 if there was nothing wrong? Right? There was something wrong. That's why people were also dying young, because there were not enough medicines. Right? So these age-related disease like Alzheimer's are coming because people are living that long. But then not everyone gets Alzheimer either. So let's not worry too much about the diseases. You live happily, you live a nice life, leave the rest to whatever it is. You do the right thing, never think. Because even if you do all the right things in life, sometimes stress comes out of nowhere, and sometimes stress disappears out of nowhere also, right? Good days come, bad days come. Just take life easily, take it lightly, do the best, and don't worry too much. And I'm telling you very seriously, it's not a thing. Don't eat out unnecessarily. If you can cook at home. Like, I know you guys cannot do that, but I'm just saying that. Don't drink alcohol, because that is really bad. Do exercise whenever you can and keep your stress levels under control. And always do something that you love. No matter how hard you work, you have to have your own fun thing going, because you have to rest when we are kids, right, we are told to study all the time, right? Just you have to do this and that. But once you are a professional, you also, when you are already disciplined enough to come to this stage at this institute here, right? If you were not disciplined, you would not be here, right? So you know how to manage your time and everything. So once you have that culture already, then you have to also find time to relax yourself in whichever way you feel relaxed. If you like reading, if you like watching something, if you like whatever you like, whichever way. So just find time to relax. Life is a lot of fun. Don't worry about it. You don't have to worry about these diseases so much. Thankfully, our diets are very good here, so that will protect us. And I will also tell you a trick, a real good trick if you don't know already. So when you cook your food, always add, if it has turmeric powder, always add kali mirch to it, black pepper. Your turmeric absorption will increase to 2,000 times. Okay, 2,000 times. That is a miraculous thing which will happen. So the cancer chances are almost negligible after that. So this is something you can always keep Kali Mirch with you, and even when you're eating in mess, you can just add. Kali Mirch will only add good taste to it, right? And your turmeric absorption will just go 2,000 times. You can go to the web and check, it is 2,000 times. So that is extremely helpful, because it will reduce inflammation in your body if there is any inflammation. It will turn off all the signaling pathways, NF-kappa B pathway, which lead to all these diseases. It is antioxidant. So all the things cancer hates, that's what it does. When you cook food at home, you guys are cooking. When you make the dough, add haldi to the dough, so that you are also consuming haldi that way. No, but you can always take it that way also. Yeah, the more haldi intake, and if you add Kali Mirch to your food in the body, whether you ate it with curry or it doesn't matter, as long as it is going in the body along with haldi, that's all it is counts.
After some, sometimes our body develop resistance toward anything, just like antibiotics, right? Is it the same case with curcumin also?
Body will never become resistant to curcumin, never ever. It's such a good thing. It will never become resistant to it. Yeah, curcumin is a miraculous thing. A lot of Indians who go to US, they spend their life working on curcumin. They spend their life working on tulsi. Yeah, all their life, like every single paper has tulsi in it, all single paper has curcumin in it. These red grapes, they are very good, right? So, red grapes, not the green ones. So, funding agencies gave a lot of money, like why red grapes are so good. The simple principle is that they have polyphenols and they are antioxidants, right? The more colorful the vegetable is, the healthier it is. Green color, red color, brown, brown unless it is not rotten. Like good colors, bright colors, good. Tomato, lycopene, right? Carrots, all good. So, blueberries are superfood. Blueberries are called superfood. Do you get blueberries now here? You don't get it, right? Yeah. I know of a person who eats blueberries, and he was telling me he gets frozen blueberries. Yeah, because it's not easy to get, and I'm guessing it is pretty expensive also. Yeah. Oh yeah, absolutely, absolutely, 100%. Yeah, Teflon covering.
It's TF, double bond TF, so it's really harmful.
I don't know that. I don't know that. If Teflon is harmful, then it is really bad. You are talking about non-stick coating?
Yeah, that
I had no idea. I would never use it if I know it, but I also use it. Yeah.
Because it releases microplastics.
exactly that I was
So Teflon is that bad?
Yes, ma'am.
Oh my God.
because it's a dimeric form.
I'm just thinking of all my utensils which have Teflon in it, and I'm not going to use it. I also have a lot of steel things, so I'm going to switch to steel things now. Yeah, thank you for telling me that. Yeah. Aluminium, like long time ago we used to use aluminium utensils, right? It was very common in old days, not recently, like even before my time, right? And aluminium, if it leaches, so especially this idli thing used to be, instead of steel, it used to be made of aluminium at that time. So aluminium, if it leaches even a little bit, it gives Alzheimer's disease. Okay, the brain degenerates. So it is really bad. Iron, we have tons of iron in our body, right? As you know, and iron is very tightly kept in check all the time, very properly kept in check. But iron in the wrong form is extremely dangerous. If it floats around freely, the ferrous and the ferric, it will be very bad. Okay, I just taught a bioinorganic course to the students, which is not even my field, but I had real fun because I learned a lot of stuff. So the metals can be really, really bad for us. Also, unless in the good form, and there is some metals which should not go in our body, like copper. We need copper for survival because there are so many enzymes which are copper dependent enzymes, like copper, zinc, sod, and all those things. But like rust is bad, and iron cannot be in the free form. So yeah, and metals can be very bad for you, and there are some metals like it is just really bad. It should not be in our body. So lead poisoning, right? Yeah. Cosmetics have contamination with metals. So there is always this, don't use Revlon, this and that, because it has this many parts. Parts per million contamination of this thing. The bottom line is that not to stress about the disease also is to keep away the disease. Not think about the disease all the time, just keep it away. It's not going to come to you. If we start stressing about the disease, we are inviting the disease, right? Yeah, so don't stress about the disease.
I think it's not just cancer we need to worry about, it's there a lot of diseases like thyroid. Recently I was reading paper, it has vast spectrum of diseases which is, so like if female gets thyroid, she's very prone to get vitiligo also.
Vertigo.
Yeah, I've seen patients also.
Thyroid is a very common disease. Like in US, I have seen every second person has thyroid thing, and I don't know. So the place where I live at Purdue, right? So when I moved there, so I bought a house, and they said, "Oh, you should put the water purifier here because the water is not pure." And I thought they are just trying to make money, and I didn't pay attention. But apparently the water isn't very good. And then I got the thyroid problem. And then I changed the water. So I buy water now. I don't drink the water which comes in the tap. So then I still take medicine, but it hasn't gone worse than before. They have the RO system, but RO system is more complicated to install. It is much easier because you have to keep changing the filter every few. But it is much better to get the water delivered at home. So you don't have to do anything. They just deliver water bottles at your home. So that is easier for us. So because we investigated the RA system, so it was just to take care of the filter and everything. So getting the water was much easier option for us. So thyroid is a pretty common problem, but it is also a very manageable disease. One thing you guys have to be cautious of, all of you, is diabetes, because diabetes is a silent killer. It's worse than cancer. So you have to really take care of diabetes. And diabetes sneaks in out of nowhere. Like just eating a little bit more sugar than usual can cause diabetes. And long-term use of this tea can cause diabetes. I'm not joking, I'm just very serious about it. That much sugar is very dangerous. So, these days I'm more concerned about these metabolic diseases because they are, in this lifestyle, obesity is increasing. Even in our country, people used to be a lot thinner before than they are now. Because there were not so many cars and people were biking, and the lives, and it will keep on changing. There's nothing good or bad about it. This is what life is. It keeps on changing all the time, right? So Initially, nobody went to gym because everybody was walking and it was a natural part of life. You know, when we used to go to school, if it was in walking distance, we would walk. We would not, take a car to go there. It was just a common culture those days. So,
Cancer causing viruses, is there any specific type of viruses which cause cancer or?
no, there are some special kind of virus like HPV and this kind of viruses which cause liver cancer. So not all cancers are caused by viruses, but liver cancer is one of the cancer which is caused by virus, and they are special virus. These are not just random virus which can cause cancer. So yeah, so that's what I'm saying, that a person is walking and just got exposed to something. Evans, go ahead.
My question is about the mRNA vaccine.
Yeah.
Okay, when you introduce the mRNA to the body, when you introduce the mRNA vaccines to the body, then
Yes, you introduce mRNA vaccine to the body.
yeah, so the cells machinery will be using it to make the proteins.
Yes.
Will make the proteins or the antibodies that be used to fight the cancer or any other disease.
Yeah.
So, and the memory will be retained in the body.
Yes.
So my question is that how is the cell retaining the memory, or how is the body retaining the memory? Yet it is starting midway. It is not starting from the DNA. It is starting from mRNA midway. So how is it able to remember?
No,
It is not starting from the scratch, the synthesis.
No, it has nothing to do with DNA or RNA there. So the way it works is this, that whether it is DNA or RNA, right? Suppose you even introduce the protein there directly, forget about the RNA, right? So if you put the protein in there, let's say for mRNA vaccine, you put mRNA there, right? So this mRNA for COVID vaccine was made against the spike protein of COVID virus, right? So they took the mRNA, they took a sequence specific to the spike, they made the RNA, they delivered it using nanoparticles or whatever they used, right? Now that mRNA will make that part of the spike protein, correct? Then what will happen is that the spike protein will be displayed as a foreign particle. So B cells will come and look at that. Okay, and the B cells will recognize, so these MSC molecules, they will present this, look at this is the foreign particle. So it is not shown as a whole spike. Spike is further made into pieces and displayed like this, right? So this is actually displayed like this. Now B cells will come and they have to bind these particles, right? So B cells will now undergo, what is that process called, like random mutagenesis in their specific regions, and they will make recombination and all of these things, and they will make trillions and trillions and trillions of combinations, and out of those trillions of combination, every guy will come and bind and see which one can bind. So 98% will die because they cannot bind, but 1 or 2% will bind those very efficiently. Those will be the survivors. And then those survivors will further tune themselves to make it even better survivors. So anything which is less will be dead. So now after like 2-3 rounds or many rounds of this, you will get B cells which bind them extremely. So they are the one which are producing antibodies, right? So those antibodies, those B cells which are producing those antibodies which are the best binders, so they will clean up the mess, and then they will, if there is no more attack there, they will retain those B cells till we live in our body, and they are called memory B cells. So anytime in the future, if a similar spike protein comes, those B cells don't have to undergo the recombination process to look for it. It's a delayed response, but it takes time. That also I'm going to tell you about the innate response versus the acquired response. So it takes few days to launch up that attack. That is why when we get vaccine, we often get fever, because the body's triggering an immune response against that vaccine. So that is when B cells are recombining, and those memory cells are always stored in our body till the last day of our life. So that is why we are always equipped. The moment we see that in a day, the whole body is full of antibodies. We clean it up. So it is retained. So you are talking about mRNA here. We don't even have to think. If we put in the protein piece, which is after there, it's going to be the same thing. If you put the mRNA in the body, it will take an hour to get the protein made. So initially before COVID, nobody thought about mRNA because they thought mRNA is going to get degraded. Nobody has ever done that. And there was a lady in UPenn, University of Pennsylvania. So she used to work on mRNA something, and nobody cared about that. They thought that she's doing a useless work. So she was thrown out, like she had a nice lab, so they took away the lab space, gave her a poor office, a poor lab. Nobody was ready to collaborate with her. Like she was given a real corner. Nobody would talk to her, nothing. And then when COVID happened, her method worked and she got the Nobel Prize. And that's when she told how she was treated by the university. So just imagine somebody really shoved down and that person gets the Nobel Prize because she was working on the mRNA part of it, which people thought was not worth working on. So science is, so even if you come up with any idea which seems pretty weird, if it seems weird to me, that is my limitation, that I'm not seeing beyond that. It is not that your idea is bad. You have to think really out of the box. And if you think out of the box, you may not know exactly how to do it, but if you think out of the box, you will find a way to do it, right? Any other questions, guys? So you guys are going to relax finally tomorrow, right? After 4 intense days. Go ahead.
We have some time.
Huh?
We have some time to entertain questions.
Oh, absolutely. I told, because I have to tell the driver to come in advance, otherwise I'm sitting here, he doesn't show up. So I told him to come at 4:15. So we have plenty of time.
Actually, I just wanted to know about drug-tolerant persisters.
About what?
Drug-tolerant persister cells.
Drug what?
Drug-tolerant persister cells, DTPs.
Oh, drug tolerant persistent cells, yeah.
Yeah, so I just wanted to know what is the highest potential research that is going on in these cells, particularly maybe in triple negative breast cancer. It is most common, the drug tolerant persister cells. So
I don't know much about drug tolerant persistent cells, but what I know about triple negative, why it is so aggressive. So there are 3 receptors, you know, right?
Yeah.
So if the receptors are expressing, you can do targeted therapy. If the receptors are not expressing, what do you target there?
Yeah,
so
exactly.
you go for chemotherapy because you just don't know what else to target, and that chemotherapy is too toxic. So a person with HER2 positive cancer was treated with chemotherapy because the person did not spend time to figure out what kind of cancer it is, and the person died. And HER2 is totally treatable with the right therapy. So that makes the triple negative cells very aggressive. So I don't know if it is the role of these persistent cells to make it aggressive. I don't know, but cancer cells keep on mutating. Just imagine if they have HER2 receptor and you target HER2 receptor, they will make HER2 disappear.
Yeah,
now
exactly.
They will depend on MET amplification.
Right.
So that is always going on. Yeah.
Yeah.
I wish I could say this is the magic bullet, but the last lecture of this Gyan course is polypharmacology. It was 10-20 years ago, my advisor coined the word magic bullet, that you have to make the magic bullet, it goes and hits the right target. Now they have realized magic bullet doesn't work. They said magic shotgun now, that you kill all the right targets at the same time, because people have realized killing one target is not going to kill cancer cells. So you just have the magic that you kill all the right people at the same time, so that they cannot bypass this to this to this to this. And that is why your question is very relevant, that figure out the resistance beforehand so that you are prepared. And these days it is very common to take biopsy from patients and you make a cell line out of that. And it is very easy to do that. So in our lab we have many patient-derived cell lines. So these are not the common cell lines. So they are better than, the general cell lines. And you can do a lot of studies there. And all these genomic, you can do single cell RNA-seq, right? You can do proteomics, you can do spatial proteomics. So you can do tons of things which will give you an idea. And these days the companies can analyze the data for you. So even if you are not so expert on it, they can help you analyze the data. And you don't have to have all the expertise in your own lab, because you send it, they will do it. So even if you have 100 different expertise in your lab, you can go and get the other things done by the companies. So that will give you quite a bit of idea of what is going on. So it's a really good idea to predict the resistance beforehand, so that you are always prepared with the next and the next and the next. All right, guys, let's wrap it for today, and then I'll see you guys on Monday.